Hello and welcome to another video in the lecture series entitled Advanced Biomaterials. In our previous videos we had seen about polymeric nanoparticles. We had discussed about polymeric nanoparticles, their important characteristics and how they can be used as delivery vehicles for targeting genes or drugs to a particular type of tissue or particular cell type. We had seen what are the important considerations while fabricating certain types of these polymeric nanoparticles, and we had also seen how these polymeric nanoparticles can specially be used as gene delivery vehicles through the formation of polymeric DNA complexes known as polyplexes. We had also seen the mechanism through which these polyplexes can enter a nucleus to deliver the target DNA. And we have also seen some of the barriers which these polyplexes can encounter during its delivery to the in vivo systems and its migration or its path to the desired site of action. So in today's video we'll be looking at some of the important preparation techniques of polymeric nanoparticles. We'll be looking at a specific preparation technique and we'll be discussing some of them and some of the others. So in the preparation strategy of polymeric nanoparticles, we'll be discussing about some of the common strategies, which are emulsification based reactions. So preparation strategies include emulsifications, and solvent another strategy is the same emulsification reaction and emulsification with reverse salting out. There is another strategy known as nano precipitation, and one of the most widely used or simple approaches for the preparation of polymeric nanoparticles include the method known as solvent evaporation. We will be discussing this method, solvent evaporation technique, for the preparation of polymeric nanoparticles to be used as drug delivery agents or drug delivery vehicles for targeting drug to a specific cell or tissue in moderate detail in today's video. So basically we have a oil in water nano emulsion in this technique. So for this one we have two phases. One will be the phase 1, which will be an aqueous solution or an aqueous phase. Now, this aqueous solution or this aqueous phase will contain water along with a surfactant. So these surfactant molecules which are frequently used, this aqueous phase will include such as ethyl polyvinyl acetate PVA, or PVA. So the phase 1, the first phase, will be an aqueous solution or an aqueous phase which will contain water and a surfactant molecule. Now there will be another phase which will be an organic phase. this will be the phase 2 or the second phase. So this second phase will essentially be the organic solution, which will contain the polymeric solution dissolved in an organic solvent. So it will contain the polymeric solution in a polar organic solvent. In this second phase, that is the organic phase, along with the polymer solution, we can add the active ingredient or the drug. So we can add the active ingredient or drug molecule, which will be included by dissolution or dispersion. So these organic solutions can be solutions such as chloroform, or dichloromethane, which were most frequently used. But since these solutions have associated toxicities, these have now been replaced by milder solutions or milder solvents such as ethyl acetate. So the polymeric solution will be dissolved in a polar organic solution, which can be ethyl acetate, and here we will be adding the active molecule or the drug molecule through dissolution or dispersion. Now what we will be doing is that we will be mixing these two phases. So the phase 1 will be the aqueous phase and phase 2 will be the organic phase. So now these two phases will then be processed or using techniques such as high pressure homogenization or sonication. HPA stands for high pressure homogenization or sonication, to give rise to a fine dispersion of nano droplets. So after sonicating or high pressure homogenization, we provide external energy to these two phases so that they can undergo this dispersion to give rise to nanodroplets in this suspension form. So these suspension of nanoparticles obtained after the so this step will be further succeeded by the step where in this this polymeric solution will be evaporated. So evaporation and purification to give rise to, and subsequent washing steps can also be prepared to give rise to these solidified nanoparticles So after evaporation of this organic phase or this organic polymer containing phase, so which can be done through magnetic shaking or in a magnetic shaker at room temperature, so this this solvent will evaporate and we'll get these solidified nanoparticles, and this can be collected by centrifugation and purified through steps such as washing, then followed by freeze drying to give rise to these polymeric nanoparticles. Now these polymeric nanoparticles, once obtained, can be lyophilized and stored for a long duration. So with this method we can see that it is a very simple and a cost-effective method for the preparation of polymeric nanoparticles, and using this method we can also load drugs as an active molecule inside these polymeric nanoparticles for their delivery into specific cells. So this is a small summary of the solvent evaporation method for the preparation of polymeric nanoparticles. Apart from these 4 methods which I have mentioned earlier, there are several other methods also which are available for the preparation of different types of polymeric nanoparticles. Now, based on the characteristics of the polymeric nanoparticles and the type of drugs also which we are trying to encapsulate or trying to coat on the surface of these polymeric nanoparticles, we can change the fabrication step accordingly. We can also change the starting materials or the polymers based on the particular type of characteristics which we are looking for in this polymeric nanoparticle systems. So with this we will be ending today's video. So in upcoming videos we will be looking at some other interesting biomaterials or molecules which can also be used as different delivery vehicles for drug delivery applications and also for other specific types of applications. So see you in the next video. Till then, bye.